Okay, protein synthesis. So this is the cell membrane. If we look out here, that's the cell membrane. And then this inside here, that's the nuclear membrane, and that's the nucleus, right? So the nucleus has these little pores in them that we call nuclear pores, and the cell membrane, or the membrane of the nucleus is so important we call it the nuclear membrane, right? Inside this nucleus, we see these red, thin strands that's called chromatin. Okay, that's chromatin. And chromatin contains DNA. DNA contains chromosomes. And chromosomes contain genes, right? So if we looked at your DNA here, and it's double-stranded nucleic acids, a gene would be a little snippet of that. So on this really long chromosome, there would be just a certain segment of it right there that would be called a gene. And that gene has a code in it. It has a sequence of nitrogenous bases that make up a code, and it codes. So the gene will code for a certain protein. A gene codes for a protein. Okay. Now all of these chromosomes are inside the nucleus. And you only have, every cell that has a nucleus, that's the only set of DNA they are given. So if that DNA is damaged, then that cell can't function. It's not going to function. So that DNA has to stay inside the nucleus, right? It has to stay in there. Otherwise, that cell is not going to be able to function. It has to be able to produce proteins. The proteins help to repair itself. The proteins help it to do its job, whether it's to create enzymes or secretions or antibodies or whatever its job is. It is doing that using the DNA, okay? They're using those genes in there. So what we see then is that DNA cannot leave the nucleus, because if it left the nucleus and it came out here in the cytoplasm, there's a lot of dangers out in the cytoplasm. There are things that will tear it down. It'll, disintegrate. It'll be destroyed. So the DNA has to stay inside the nucleus, right? So we're going to talk about two processes. One is called transcription. One is called translation. And those are the two processes to produce a protein. The little grayed area that you see right there, that's the nucleoli, or nucleolus is singular. And that's what produces the ribosomal parts and the ribosomal RNA. So it's basically producing the ribosome. And then the ribosome comes out to the cytoplasm, and then we see the little ribosomes all over the place. These are what ribosomes are. Okay? And that's where the protein is going to be produced. Right? All right, so this is going to be a very simplistic explanation of what happens, but okay, I'm just going to erase this stuff so we can take a look at what's going on inside the nucleus. Now that you have all that. All right, so inside the nucleus, we have a DNA molecule. There's our DNA molecule. It's the book of life. It helps that cell to function, to live. It cannot leave the nucleus. So just like a book in the bookstore, or not in the bookstore, but in the library, a book in the library that you don't want destroyed, but you want some of the information, you have to take the information out of the library, but you can't take the book out of the library, what would you do? You'd make a copy of it, right? So you'd make a copy of it. So that's exactly what happens. We make a copy of that DNA molecule, and the copy is called RNA, and it only has one strand. Has one strand. So now we have an RNA molecule, has one strand of nucleic acids, right? That process right there is called transcription. You're transcribing the DNA into the messenger RNA. So that's transcription. Transcription. Now that messenger RNA molecule has a couple of things that are nonsense that we have to get rid of, and those are called introns. So we have to edit the RNA. And this is actually, the name of the RNA is messenger RNA, so we put a little M in front of it. That's messenger RNA. Now, messenger RNA has to be edited, so that's exactly what happens. We remove the introns, the nonsense things, and now we have a strand of RNA that's ready to leave the nucleus. So we edit, and we remove introns. Okay? And now we have a messenger RNA that's ready to leave the nucleus. Now, it can leave the nucleus through the little pores. So the messenger RNA leaves through the pore, and it goes out to the cytoplasm, and it attaches itself to one of those little ribosomes. Right? So now we have a ribosome, and we have a messenger RNA on there. Now, this RNA is made up of a strand with these nitrogenous bases attached to them. And so there's only 4 bases. Adenine, I don't know if you guys remember this, adenine, uracil, cytosine, guanine, right? Do you remember those? So there's only 4. But those 4, those are the 4 types. They can be in numerous amounts, and they can be mixed up in different orders, and that's what would actually code for the protein. So we have this messenger RNA out here, and it might have it'll have a very specific order and number of those bases. And so those bases, we usually label them by their first initial. So we would say that it had, say this order would be cytosine, guanine, and adenine. So say it's like in this order, right? I'm just making up like an order of bases. Uracil, cytosine, guanine, adenine, cytosine, guanine. That's just a specific order and a specific number, right? On that messenger RNA. And it's coding for something. But the cell, the cytoplasm of the cell, it says that's gibberish to me. I do not understand bases. Don't understand bases. So another process has to occur, which is called translation. And so during the process of translation, this messenger RNA gets translated into a polypeptide, which is just a long line of amino acids attached to each other. Now the cell knows what to do with a polypeptide because the polypeptide is basically a small protein. That's what it was trying to make. So that process of the messenger RNA getting translated into a polypeptide is called translation. There's a process and there's steps, but I really don't need you to know the steps. There's a few things I need you to know though. For instance, like transcription. Where does it occur in the cell? Well, it occurs in the nucleus. And what's the end product of transcription? Well, you get a messenger RNA, right? And then if we talk about translation, where does it occur in the cell? Well, it occurs in the cytoplasm. And what do you get? What do you get from what to what? You take a messenger RNA and you are going to make a polypeptide out of it. You're going to translate it into a polypeptide. And we don't need to go through the steps. That's more of a biology class. It's not our A&P class, so we're not going to go through all of those steps. Okay?
So the adenine and cytosine and guanine, those are all
Bases. Those are all,
nitrogenous bases?
they're nitrogen bases or nitrogenous bases. Sometimes I think the longer story helps you to remember it better, but I don't have time to go through the longer story.
Right.
So this is, and so really, if you have a cell, there are cells in your body that don't have a nucleus. Like red blood cells, they do not have a nuclei. So they cannot make proteins. And they can't make proteins to repair themselves. So what do you think about them? They're probably not going to live long, right? And they don't. They live 120 days and they're dead. They can't repair themselves. Then there's other cells, like some of your white blood cells. They live for 20 plus years because they have a nucleus that can repair themselves. Your muscle cells, whatever muscle cells you're born with, that's all you have the rest of your life. So they are going to repair themselves. They have nuclei and they're going to repair themselves over and over again, right? Now, in addition to that, then, we have, in addition to the nucleus, if the cell has a nucleus, it can make proteins. That's key. If it has a nucleus, it can make proteins. In addition to that, if it has a nucleus and it has those little centrioles, then it has the ability to divide. So some of your cells have a nucleus, but they don't have centrioles, like your neurons. There's no centrioles in your neurons, right? So they can make proteins, but they can't divide. So whatever neurons you have, that's it, Okay, so that is my talk on transcription and translation.